Thank you for watching Modality Therapeutics Business and Financial Report of the second quarter 2026. Here I am making the forward-looking statement. So for those new to Modality, let me go quickly through the corporate overview. The Modality is a biotech company established in 2016 around the IP of CRISPR technology. We are headquartered in Tokyo and listed on the Tokyo Stock Exchange market and operated in Boston, Massachusetts, United States, with around 20-ish people. So what we do with our technology is essentially turning on beneficial gene and turning off the harmful gene so that we can treat the genetic disorder with epigenetic editing technology. Here is our value highlights. First, we are the first company advancing a CRISPR-based gene activation therapeutics toward the clinic. And we have 4 programs across neuromuscular disorders. And also we have a multiple therapeutic target validated in vivo. Our lead program, MDO101, is making progress toward an IND. Our competitive advantages include gene regulation technology without cleaving DNA, unlike the gene editing. And secondly, we have demonstrated long-term gene regulation. And thirdly, this technology is applicable to neuromuscular to CNS disorders. And we have established tissue-selective AAV delivery with the collaboration, throughout the collaboration. And lastly, this technology is protected by robust intellectual property portfolios. Right. This slide is a schematic representation of a CRISPR GANDAM molecule, which is the abbreviation of a guided nucleotide-directed modulation system. So the molecule consists of 3 components. First, guide RNA as pointer, and the second, dCAS9 as binder, and then the thirdly, epigenome editor that turn on or off the gene specifically. So by combining this molecule, we are able to bring this molecule to the specific position of the chromosome so that we can turn on or off the gene specifically. So we put this molecule in the form of DNA and pack it in the AB vector. So then the AB, we use a tissue-specific AB so that we can bring this molecule to the tissue-selective manner. And then this GANDAM molecule called in DNA form is translated into a protein under the regulation of tissue-specific promoter. So then the translated molecule, the protein, GANDAM molecule, and the guide RNA binds to specific position in the tissue, in the target tissue, and then turn on or off the gene in the selective manner that leads to the efficacy. Our core competency is the ability to identify and optimize the GANDAM payload that is protected by IP, and we have the ability to select the right target for this technology, and we actually identify and optimize the right molecule that to be developed. And also we have ability to select the best capsid in the field so that we can combine our payload with the best vector to make the optimized gene therapy for each target. And the benefit of that is 4 points. One, we don't alter or we don't create the DNA, so the DNA remain intact. So it's important in terms of the safety. And the second, This requires just a single dose, and that leads to the third point, long-lasting efficacy. And the efficacy is not is a transformative and a disease-modifying efficacy. This slide shows gene therapies approved in the United States. As shown here, starting in 2017, a number of gene therapies have been approved in the United States for multiple genetic disorder. And this slide shows the competitive landscape in the field of epigenomic editing. So when we started epigenomic editing, we are the only company doing so. However, a number of companies listed here are following us with the similar technology. But what made us unique, even in this field, is that we are the only company primarily focused on epigenome activation rather than gene suppression, in the belief that the silencing of the target can be achieved by not just by epigenetic editing, but by multiple modality, like an antibody at the level of a protein, and also RNAi or antisense oligo technology. Whereas activation side, maybe a very limited technology can do so. And also, by the reason that I'm going to tell you in the following slide, certain disorder can be treated only by epigenetic editing. So we focus on the disorders that cannot be achieved by the treatment cannot be achieved by any other technology, like MDL 101 for RAMA2-RD. So with the following slide, I'm going to tell you the status of the business, followed by the financial update and growth strategy and the summary, followed by the Q&A. Let's start with the status of the business. And for this time, I am primarily focused on the status of MDL101 because this is the crown jewel of our pipeline, and this is a real important validation of the technology. And for this quarter, we are able to share you that very clear timeline toward the IND as a result of the recent achievement. So here are the 3 points I'm going to share with you in this presentation. The first, the progress and timeline of MDL101, and the second, the progress in the other programs, and thirdly, the financial, the other progress in the business. Just to recap the pipeline of the company, as shown here, our focus is the muscle disorder shown in the blue, and including the RAMA2, the MDL101 for RAMA2-RD. And also we have a pipeline for dilated cardiomyopathy or heart disorder, and also the 3 pipeline for CNS disorders as well. Okay, just briefly explain what MDL101 is and what the target indication is with this slide. So RAMA2 related disease is very severe disorder caused by a mutation in the gene called RAMA2. And the RAMA2 gene codes one of the 3 protein that forms the laminin complex shown in the left bottom of the slide. So the mutation or deletion of the gene leads to malfunction of muscle. And this is a very rare disorder with the prevalence of 8.3 patients in a million population. So around 2,000 to 2,500 patients are affected in United States. So on this slide shows why this this disorder need to be treated by the epigenome editing technology. So the disease-causing gene, RAMA2, is a large size gene with a size of 9 kilobase pair. So this cannot be packed in single AAV, right? So the conventional gene therapy is not applicable for this disorder. But interestingly, this RAMA2 gene has a sister gene called the RAMA1, as shown in the left-hand side. The structure of the molecule is very similar to RAMA2, and also it has been proven that RAMA1 protein can compensate the function, most of the function of RAMA2 in disease model. But this RAMA1 gene is intact in most of the patient and most of the people. However, in muscle cells, this gene is silent, as shown in the mid column. So what we can do with the Gundam technology is to activate this silent RAMA1 gene in muscle so that the translated protein can compensate the missing function of RAMA2 in the patient. And actually, that worked as we hypothesized. And this slide shows the outcome of the disease model we just completed in the IND enabling mice study. The left-hand side is the survival curve of this mice. And this mice, like patient, lacks the complete RAMA2 gene in both alleles. So as you can see here, the mice die relatively quickly, and the mice do not survive beyond the 300 days, as shown in the blue line, right? But the disease model mice treated with the Gundam molecule, as shown, show the much longer survival, shown in orange, green, and the purple line. This represent low and mid and high dose of the Gundam treatment. So, and the mice group treated with mid and high dose has over 90% survival even after 300 days. So this is a significant improvement of the survival. And secondly, I wanted to show the improvement of the function. As shown in the mid picture, this is the improved grip strength of the mice of the four pounds, right? As you can see here, we are able to demonstrate the dose-dependent improvement of the grip strength. It doesn't reach to the wild-type mice. However, as shown here, it's demonstrated a dramatic improvement of the grip strength. And also shown in our right-hand side, the body weight that partially represents the recovery of the muscle is improved in the dose-dependent manner. And also on the right bottom, the health deficit is also improved in dose-dependent manner. So, including this, we are able to show you, demonstrated the efficacy of the molecule in disease model. So we are very much convinced with the efficacy data. So this slide shows the key milestone advancing towards the IND submission, including the mice IND enabling data as I shown in the previous slide. So first, we have a very robust efficacy data in the mice IND enabling study. So we are convincing to bring this molecule towards the clinic, right? So that's the first point. And the second point is that we also run additional NHP study to validate the pre-treatment protocol at multiple CROs. But with that data, we are able to optimize the pre-treatment protocol of the protocol, and that has been clarified a mitigation strategy of the product. And thirdly, Execution plan toward the IND, including a GMP manufacturing. This is another important piece for IND filing. So the manufacturing includes the plasmid GMP manufacturing, which is ongoing, and also AB GMP manufacturing. We are starting in this third quarter. So we have a clear view that when this will be completed and when we can file the IND with the GMP manufacturing and also the additional GLP tox study and also the alignment with the clinical side to get ready for the clinical trial. So as a result, in summary, the MDO-101 is on track to submit IND in late 2026 or first quarter of 2027, with the goal of advancing toward the first in-human clinical trial in the United States. So this is another view of how the timeline looks. So we have a 4-work stream toward the IND. First is the pharmacology, which is almost completed, and also non-clinical study, followed by additional validation in the NHP study. We're going to run additional NHP study that will be a part of the IND document. And the thirdly, GMP manufacturing is ongoing and we'll be able to complete it by the end of this year, right? And also we have a discussion with the clinical candidate side so that we can jumpstart the clinical study once we get the IND clearance from the FDA. So this slide shows how our clinical trial design looks like. So essentially, we have 2 cohorts. First cohort is the low dose cohort with the patient number 3, and also we have a high dose cohort with the patient of 3. And what we are trying to achieve this clinical trial is first to demonstrate the safety, and also we wanted to see the efficacy by comparing the outcome of the data with the ongoing natural history study shown in the bottom of the slide. So we have our clinical trial design and also the readout is adapted to the natural history study. So we'll be able to compare the outcome with the natural history study to see the efficacy there. The inclusion criteria of the patient are the patient under the 5 years old and the secondary genetically confirmed by allelic, pathogenetic, or likelihood of a pathogenic LAMA2 mutation, and a severe LAMA2R clinical phenotype, and also AV9 antibody negative. This slide shows the development priority and key milestone. Again, our primary focus is MDO-101 in the belief that this molecule is not just the validation of the molecule itself, but also the validation of the technology. And also, this is development of MDO-101 is the paving the way for the following program, particularly the muscle disorder, muscle program that shared multiple components. And also, this will open up the opportunity for larger indication like a DMD, DM1, and FSHD. So let's look at the key milestone for MDO-101 first. So as I mentioned in the earlier slide, we are expecting to file IND late this year or early first quarter of 2027. And we are hoping to achieve the first patient dose first half of 2027. And probably we'll be able to see the initial clinical data later of the year. And also the first half of 2028 we are hoping to have established a clinical proof of concept. And with that data, we are aiming to file the conditional approval in the United States. So by going through this path and by establishing proof of concept with MDO-101, the pipeline expansion opportunity is open for other disorder like MDO-201 for targeting DMD, 202 for DM1, and also 103 for FSHD. So this slide shows the overall pipeline update of our entire program. I'm not gonna go through the entire slide, but as I mentioned in the earlier slide, MDO-101 is moving towards the IND, so that's a big thing. And also, we are in development of MDO-201 for DMD, 202 for DM1, and 103 for FSHD disorder. So these three programs are in the preclinical stage, but in collaboration with the collaborator for each program, we are trying to develop this molecule toward the IND after the MDO-101. So the data has been presented at multiple occasions, including SGCT, and we presented the DM1 data there and also FSHD data at SGCT and also FSHD Society Congress. So we are making progress not just in MDO-101 development, but also the following programs include primarily in the muscle disorders. Okay, in the next part, I'm going to give you an update of financial status. And at the end of the second quarter, we have a cash position of 3 billion yen, or about $9 million US. So this cash position allows us to develop MDO-101 toward the clinic, IND enabling, and also beyond. So we are in the good position to fund the READ program. And on the PL, profit and loss side, we spend about 1 billion yen in the first half of the year, or about $6.1 million US, including 5.6 million R&D cost. So as shown here, the primary cost is buried in the R&D to bring our MDO-101 program toward the IND. And we are expecting to spend about the same amount in the second half of the year. So the second half of the year. Okay, so on the financial side, obviously, the development of MDO-101 remain to require the funding. So we continue to fund this program with the cash position right now and also additional finance. And then, as shown on the bottom of the finance slide, we have an ongoing warrant-based finance in collaboration with the EVO Fund. So we have allocated 240,000 unit or 200 million share equivalent warrant to EVO Fund, and the EVO Fund is exercising it stepwise manner. So this cash will bring us to achieve the IND and also proof of concept of MDO-101 first, and because this is important to establish platform validation that open up the opportunity for the following program like MDO-201 for DMD and FSHD and also DM1 program as well. So what we are trying to achieve with the current cash and the cash we are raising is that first, clinical proof of concept of MDO-101 and also validation of the Gundam technology. And also we are investing in the following program to maximize the growth opportunity with the following program. So this will able to realize the path to a conditional approval of MD-101 in the United States and also other country. And hopefully we might be able to establish the potential in-house sales. I'm going to show you in the next slide. And this will increase the likelihood of success of the subsequent large-scale, large indication program like MDO-201. Okay, in the following few slides, I'm going to show you the growth strategy and where we are and where we head to. As I mentioned in earlier slides, our primary focus is MDO-101, and by developing this to the higher stage, we can increase the value of the pipeline, along with expansion of the opportunity towards the other indication with the following program like MDO-201 and 202 and 103 for FSHD. So with the combination of those will maximize our growth potential in 2 dimensions. So at this moment, we have a multiple constraint in resources. First, we have a limited capital, and the capital is good enough to cover MDO-101 cost, but not enough to cover the development of the following program. So that's the limitation. And also we have a limited development band-wise. And thirdly, we only have one lead program in the pipeline. And also CNS delivery has not yet been matured. So that's the current limitation of our development. However, once we establish proof of concept with MDO-101, this will release the constraint right now. And first, we are able to achieve the technology validation. So we have a much higher confidence in the technology. And that will enable us to get easier fundraising partnering. And also this accumulated know-how is reusable for development of the following program because most of the program share the common technology. And also this open up the opportunity to develop the other program much faster, much efficiently. So this will also open up this will increase the value of the company. And also by the time that we establish proof of concept in MDO-101, the expansion, the maturity of the CNS delivery is hopefully be achieved. So we can combine the better AAV delivery toward the CNS that go beyond the blood-brain barrier. So we are able to develop the program for CNS disorder much efficiently. Okay, so in this slide, I'm going to show you the opportunity of the value creation just with MDO-101. But also we have a much upside opportunity with the other program. But just to focus on MDO-101 opportunity. So we have a main scenario. One is the direct sales, and the other is the partnering at certain point. So direct sales can maximize the future sales, which can be pour a reasonable amount of money into the corporate portfolio. But the downside is that we have to bear development and sales costs in-house, and also we have a significant need for additional fundraising. On the other hand, partnering can secure non-dilutive funding at an earlier stage, and also the development and the services can be shared. However, the downside is that the future revenue is limited to royalty or milestone. So in the case of direct sales, we are hoping to get a US accelerated approval around 2018 to 2019. So that will bring us the sales. Let's say we dose 10 patients with the drug price of 3 million, we are able to achieve 3 million sales through it. On top of that, as we get approval from the US government, we'll be able to get a priority voucher for our rare pediatric disease designation. So currently, the RPV is traded at around the price of 200 million. So we'll be able to, we are hoping to trade the PRV to the other company, and we are able to earn that amount of money at a pure cash income. So, and then not just the first year, in the following year, we can make the sales along with the dosing to the patient. So we have a significant amount of the sales income along with the sales of MDO-101. Oh, so in the case of the partnering, our income is limited solely to the royalty and a milestone. So let's say that we get royalty rate of 20%. So in such case, when we dose to the 10 patient and the sales is around 30 million, so our income is around 6 million for the first year, and also 12 million for the 20 dose and on the second year, and so on. So I can't tell you which is the better future for us, because it's a condition. However, but as shown here, once we get approval, this will open up the opportunity for us to fund the more program by ourselves throughout the sales we are achieving through the MDO-101. Okay, so in summary, the key takeaway of a second quarter 2026 report is, first, we have completed disease model study of MDO-101, and we have established very robust animal proof of concept data, including extended survival and improved muscle function. And the secondary, we conducted and completed additional validation of the study in NHP, and this support the rationale for a clinical trial, and also we are able to improve the visibility of the IND target, which is going to be end of 2026 to first quarter of 2027. And then lastly, the 19 series of stock option or warrant-based finance is allocated to granted to investor, and the exercise is underway that support our development of MDO-101 and advancement of following program in progress. Okay, so with that, I wanted to finish the presentation of the second quarter 2026, and maybe we put the Q&A at the last part. I'm not gonna go through it, but I hope that this will improve your understanding on where we are and we are heading to. Right. Thank you so much for attention, and see you in the next occasion. Thank you.